Hi, my name is Adam Hornbaker, and I worked in Dr. Gonder's lab this term and was mentored by Miss Dana Mitchell. Before we dive into my project, it is important to know that Cameroon is home to three genetically distinct populations of chimpanzees that are genetically diversifying. They have measurable genetic differences from each other, but how they became genetically different isn't well known and could be in part due to the theory of natural selection. The three aforementioned populations live in three distinct habitats: one Guinean rainforest, another Congolian rainforest, and the third in a savanna woodland mosaic ecotone habitat. Since they all live in different habitats, it is safe to assume that they are exposed to different stimuli which can influence their preferences, culture, as well as their genetic makeup, as we will see later. One of the results of living in different habitats is that they have slightly different diets from one another. By collecting fecal samples from the environment, it has been observed that chimpanzees in the Guinean rainforest and ecotone habitats have different food preferences from one another. For example, ecotone chimpanzees appear to sometimes have a higher volume of animal parts in their diet as compared to Guinean rainforest chimpanzees, as indicated by the vectors in Figure 2. This observation leads me to my research question, which is: does the observed variation in chimpanzee diet have genetic implications as a result of natural selection? More specifically, I want to try and see if there is any evidence for differentiation or separation in the frequency of genes that have to do with diet across populations, which would lead me to the conclusion that natural selection is taking place. In order to figure out if natural selection could be occurring, I first had to determine the function of genes that were associated with single nucleotide polymorphisms, or SNPs, that I was provided with. The gene functions I paid special attention to were the ones that had some sort of dietary consequence when expressed. According to KEGG, the gene ACAT2 functions in lipid metabolism by synthesizing cholesterol esters which help to form chylomicrons. The job of the chylomicron is to transport lipids to other parts of the body where they are broken down and absorbed by tissues. Since ACAT2 is involved in lipid metabolism and is a diet-related gene, it would be useful to plot the SNP that is close to the ACAT2 gene to see if there is any differentiation. Plotting the frequency of the SNP near the ACAT2 gene against temperature seasonality, an environmental variable that varies across the three populations, did indeed result in observable differentiation, as you can see in the spatial analysis in Figure 4, because the frequencies across the populations vary. To further back this up with statistics, the linear regression model in Figure 5 shows that the SNP is significantly correlated to temperature seasonality with a p-value of 0.000892 and an r-squared value of 0.5574. Overall, these findings provide evidence that natural selection could be acting on the region of the genome around the ACAT2 gene. To touch back on my research question, which asked if the observed variation in chimpanzee diet has any genetic implication, the results show that yes, there could be genetic implications since there is differentiation in the SNP associated with ACAT2 across the chimpanzee populations. I theorize that this may contribute to natural selection by allowing the propagation of behavior associated with consumption of lipid-rich animal products in ecotone individuals, since Figure 2 shows us ecotone individuals having a dietary preference for animal products which have a lot of lipids. However, in order to confirm decisively if natural selection is acting on the ACAT2 gene and not just on the other genes in the area, future work would involve the sequencing of genes and subsequent alleles and observing their differentiation. Overall, this project shows how genes are at the mercy of the environment, so you can see how man-made disturbances would drastically impact a population living in their habitat. This project and a project like this could help with conservation efforts. A critical habitat is defined by the Endangered Species Act as an area where conservation is needed in order to preserve the endangered species that reside there. Knowing this, research that investigates genetics could help with the identification of critical conservation habitats, since they help bring light to components of the genetic makeup that convey to us what populations need in order to survive and thrive in their environment. For example, this project shows that ecotone chimpanzees could be genetically wired to be able to break down foods that contain more lipids than other populations. So, conservationists can mark up the ecotone habitat range as an area that can't be hunted in or around by humans, since that could eliminate animal foods that the ecotone chimps rely on so much to survive. This concludes my presentation. Thank you so much.